Okay, that was quite a tour de force of the entire field. Thank you, everyone. So we'd like to have a discussion now, so if I can invite the speakers up here so you can be grilled. So Christine, Jeremy, and Travis, thank you. So we wanted to give the opportunity to have a little bit more of a discussion because we know after each of the talks you're probably overwhelmed by the amount of data that you've just seen, and this is quite a rapidly growing field. So I wanted to give people in the audience an opportunity to ask any questions that you may have that arose from the original talk. Maybe I'll start by asking a question of Jeremy, of course, just because it's fun. Jeremy, can you go into a few of the antigens that may be the targets of Zika virus? Do you have an idea of why they may be more specific to GBM stem cells? No. Everybody wants coffee, so this is going to go well because we're going to get through this quickly. All right.
It's an area that is highly controversial, and we've been looking at a number of the receptors that have been proposed as receptors for the normal neural stem cells. It's still not definitive, but obviously things like AXL and TYRO3 and others have been thought of. Musashi 1 is an area that people have looked at, and so we've been looking at it. The specificity, one of the challenges is that cancer stem cells are not as distinguished by single antigens or markers as the field has often suggested, and so my suspicion is that it's a little bit more complicated than just a single antigen, but we already know the inflammatory response is one of those.
Can you use Zika as an imaging agent?
So that's a great question. So one of the things we're doing is, so obviously you want to kind of track down all the stem cells, especially because the progeny of the stem cells are the ones that are invading into normal brain areas. And so we've been using phage display, and I suspect in combination with the Zika approach that we could get a theranostics approach. So that's a great question.
Very nice. Allison.
It is a related question. So the Zika virus also infected postmitotic cells, in fact neurons, in fact astrocytes. You not only have the progenitor cells in the brain, but all those glial progenitors. So how do you see, I know it's early days, early steps, and I find it ingenious that you are thinking about those ideas, but how do you see the future? Would you engineer the virus? What is the future?
So we're in collaboration with Michael Diamond and Milan Cheta at WashU, and so what we're doing is using different mutant forms. So there are two different approaches we're trying to take. One is to leverage some of the work, for example, you and others have done, and find drugs that actually perhaps protect normal cells but not cancer stem cells. So, and then in addition use drugs that sensitize the cancer stem cells. So what we want to do is widen that therapeutic index, get some efficacy, but also using mutations of the Zika virus that have some aspects that may be less toxic against normal. And so we already have the next roundup of mutants that we're going ahead and testing. So I think a combination of genetics and pharmacologic aspects could really be very exciting.
Okay, I had a question for Christine before anybody else has questions. With the GBMs, the blood-brain barrier is a little leaky. Have you tried IV administration of your CAR T, or is that something you anticipate could be done in the future, targeting IL-13 receptor alpha?
All right, so, I think when you think about route of delivery, it's much more clinically feasible, one might argue, to give it systemically. And so it's known that the blood-brain barrier is somewhat leaky. Our preclinical data really suggests a benefit on a per-cell basis going locally, and the device is very well tolerated for patients. And I think a lot of therapies are given intrathecally or into the CSF, and so we're moving forward in that way, although others are looking at systemic delivery. I think we would have to change our platform and how we delivered the cells, because we would probably have to start thinking about a lymphodepletive regimen prior to administering if we went systemically. Some of our other platforms, we're opening a trial for HER2 and treating women with breast-to-brain metastases. And there, when you have systemic disease, I think that might be the platform where we really rigorously compare local versus systemic delivery.
There's a question in the back here.
Actually, I have a question for you, Kat. You mentioned that at the end of your talk, you mentioned a relationship between IL-6 receptor, and ADAR. I was wondering what will happen if we have sudden high expression or high secretion of IL-6.
So if you have very high expression of IL-6, this is an inflammatory cytokine that's released in GBMs and many other tumors, whether it's myeloma or heme malignancy. That seems to activate ADAR probably the most robustly. So that could be a good thing unless it's turned on sustainably. So we really see these RNA alterations that are really cancer-provoking when it's turned on at very high levels and sustainably. So I think we just need to dial it down like the thermostat, which is sometimes too low in this room, I'm just saying. But we can dial it down with JAK2 inhibition. We're trying to work on a selective ADAR inhibitor and see if we can get away from that approach of just using JAK2 inhibition. But as you can imagine, our stem cells need ADAR to self-renew and also to protect themselves against viruses that have an RNA-DNA intermediate. So if we're too good at inhibiting ADAR, actually we won't have protection against retroviruses like HIV. So it's going to be a pretty measured approach. But thanks for the question. Anybody else? Yes. Pat Olson from CERN. There you go.
Is there reason to believe that CAR T therapies will be more durable in response, a single antigen targeted? And you may have alluded to this at one point when you said that it may have generated a more robust, broader immune response. I think one of you mentioned that. But I guess, many of us have seen targeted small molecule therapies, Gleevec and their followers. And I was just curious if some of you could discuss, because I know Kat's always talking about combination therapies. So I was curious if there's reason to believe that these would be more durable.
So to step back, I think the success in CD19 CAR T cells is because we have persistence and proliferation. So early trials, City of Hope was the first to treat patients with CD19 CARs. The problem is they only persisted for a day. So they weren't durable at all. And so now we start to see persistence in B-cell aplasia a month to 3 months, although in some patients there's relapse. So in that setting, we learn by looking at B-cell aplasia as a surrogate marker, and also the durability of the therapy, that the goal is to have these cells persist. And so as we improve manufacturing, as we improve CAR design, I think it's reasonable to think that you'll have a resident population of memory cells that will be available to target antigen or the cancer when it comes back. The issue is immune escape, and so how do you address that? And our unique responder, I think it's been very informative. I think not only did it evade because it lost the target antigen, but it also, we believe, lost neoantigen expression, and the tumor became, lost antigen presentation machinery and other things that are studies that are sort of ongoing. But I think you need both aspects.
I think that's right. And I think when you get expansion of a memory population, all these CARs still have TCRs, all these CAR T cells. And so if you can engage the TCR as well, you can get antigen spreading from that effect if these are a persistent population. And then the CARs can be more of a direct mechanism of cytotoxicity, but also an adjuvant for the immune system.
So Christine, when you talk about immune escape, do you think that those are clones that are pre-existent, or is there plasticity of the system?
So I think it can be both, but that's just my opinion. So I think absolutely when you have a heterogeneous tumor, you're going to have clones that grow out. So what's interesting in our one case study is none of the original lesions ever recurred, not adjacent. They were completely new areas, new lesions. And so there were truncal mutations that were similar, so we knew it was the same cancer, but it really makes us think that there was some seed somewhere that then initiated a new tumor locations.
So speaking of targeting the seed, just one more point to get back to Pat's point. I think that we can work together to combine these immunotherapies, switchable CARs, your CARs, Jeremy's Zika virus, and then actually using self-renewal antagonists that we already know have a therapeutic index. So, for example, a hedgehog antagonist we've already used in clinical trials, JAK2 inhibitors. So I completely agree with you, Pat. The multi-pronged approach, especially when we're dealing with draconian diseases like this, may be the way to go. We have a question here.
Did you see any phenotypic differences in the glioblastoma hot tumors? Was there more of like a microglia response to that? And do you have any hopes of attenuating those tumors to create more of a response?
Right, so those studies are ongoing right now. We're really interested in interrogating the panel of patients we're treating and what the microenvironment is at the time of treatment and post-therapy. And so those are ongoing. I will tell you that different patients respond in different ways. We see increases in T cell populations and other immune infiltrates into the CSF, and they change. And there's those that we think are promising and those we think that indicate progression. But those are absolutely is what the sort of things we're thinking about.
All right. Any more questions? All right. Okay. And then, Travis, I had a question about the switchable CAR technology for splice isoforms. I know we've talked about this, but how readily do you think you can target splice isoforms considering that splicing occurs so much in cancer?
Sure, and I think anything that you can develop a therapeutic antibody to, we can develop into a switch molecule in a similar fashion. You can also think about what we've done is studies related to the question on whether or not the antigen negative cells exist at the time of treatment. We've done studies where we've looked at titrating in the CD19 negative Raji cells mixed with the CD19 positive Raji cells to understand what's the limit of the least number of cells you need there in order to cause a relapse event, at least in these models. And so we can mix switches, or we can develop, for example, with CEA, where we develop switches for antibodies that have more membrane proximal domains that are less likely to be inhibited by shed antigen or isoform variants. I think there's a number of approaches where we can do that, not only in the single targeting agent, but also in using mixed switches at the same time. So I showed that was a sequential targeting, but we can also do the simultaneous.
Very nice. Yeah. Well, I'd like to thank everyone for their attention, Luigi for a fantastic talk, and our three speakers for following up and taking the baton and running with it. I think Jennifer Braswell deserves a lot of credit for being a sponsor of this meeting through the Stanford Stem Cell Clinical Center and directing us together with Larry to have these meetings. We get a 15-minute break now, so thanks everyone.